On to the difference between gene and cistone. As in class I have told that what is gene? It is the segment of DNA that synthesize any kind of RNA. Like see, this part is synthesizing rRNA, mRNA, tRNA, sRNA. So the DNA part of the segment of DNA that synthesizing any of this RNA is called a gene. So these all are gene as synthesizing any of the RNA. But cistone is that the part of the segment which is only synthesizing mRNA. So initially it is synthesizing only the protein is called the cistone. So see here the segment of the DNA which is synthesizing only the mRNA is or I also can which is synthesizing the protein is called the cistone. Now let's see the difference between monocystonic and polycystonic. What happens in the monocystonic is actually present in the eukaryotes and polycystonic is present in the prokaryotes. From you can name itself, you can see polycystonic means the many protein and monocystonic means the only one protein. Thus what happens in the eukaryotes that the only the segment of the DNA which only synthesizing the mRNA, uh, and then it is only synthesizing only one type of protein, single protein. It is called a monocystonic. One cystone, one gene equals to one cystone here. Okay, but here the segment of DNA, there are many segment of DNA thus initially producing many kinds of protein. Like there is segment A, B, C, all the here in case of prokaryote all of the genes are being transcribed at once. They are not like means all of the transcribe all of the segments are getting transcribed at once, not like in the eukaryotes that only one segment will get transcribed on in one time. No, what in prokaryotes all of the segments are getting transcribed at one and thus producing more than one protein. Thus it is called polycystonic in this case. Okay, then it comes the intron and the exon part. Monocystonic genes in the eukaryote, here you can see that this is the cystone or the gene. Okay, so here some of the parts are exon and some of the parts are introns. So introns are those parts which are not being transcribed into protein and the exons are the parts which are getting transcribed into the protein. So the introns what here is interfering with the protein sequence. Okay, protein sequence, like you here can see that AUG is there, then UUG something or any other protein codon is there which is getting transcribed into protein, but here the coding, the nucleotide sequence is not transcribing into any kind of protein. So they are actually interfering with the sequence of the protein. We don't need this part, so what you have to do, you have to remove this part. So introns are those part we have to remove in the post modification, or I can also say the post modification are those processes which are required to filter the mRNA. Okay, so after this is the mRNA and this is the mRNA, and I don't this is good, this is a sequence of the DNA segment of the DNA which is getting mRNA. In that mRNA there be exons also, introns also. So introns I don't need and exons are the parts that we need. So see, exons are the expressing segment and introns are the interfering. They are interfering as it is interfering the exon, so it is also called interfering the intervening segment. Introns cannot be removed from the DNA. Okay, they cannot be introns. So what happens, we have to remove from the mRNA. We can't remove introns and introns from the, but we have to remove the introns. So what we do, we don't remove the introns from the DNA. What happens, first we transcribe the DNA into mRNA, then in the post transcriptional modification we remove the introns and the exons are get attached to each other and thus new protein, new mRNA is formed. Okay, new mRNA is formed.